Hi, everybody. I'm glad you're here. I've been asked to talk a little bit about the work that we do in my lab, but I want to ask you, why should we study paleontology? Why is it important to study things that lived and existed on our planet before us? This has often been thought a relic science, something that really doesn't contribute a whole lot to what we know and where we're going. But I would argue just exactly the opposite. Why should we study the ancient past? Well, if you want to predict anything about anything from where your budget is going to medical outcomes to how drugs work, you need to study the past because all prediction is based on history. Every last little bit of it. You cannot make a prediction without the past. If it's the future that concerns us, our future, then we only can look backwards in time. And that's because pretty much all available data is locked in the rock record. So if this is the history of our planet, you have 4.6 billion years where the Earth began and virtually nothing happened except the evolution of microbes. Finally, at about 600 million years ago, we began to see complex life. So the vast majority of our planet, there's been no vertebrates, there's been no anything really that we can study. All of this lifespan here, the only record we have is the fossil record. And if we want to make predictions based upon human history, we just have this little teeny tiny corner of the planet where humans were involved to make those on. We need the rock record. So today, if we want to study organisms and how they fit in their environment and their unique adaptations, we can use the tool of genomics and proteomics. We know that if you have a DNA sequence, it has direct impact on an organism that was produced by it. So DNA influences all kinds of stuff from how tall you'll be and how you digest your food to what color your skin and hair and eyes are. And when we began to realize this relationship between DNA and organisms, it brought about a molecular revolution in how we study biology and evolution. We do it very differently than we did 100 years ago. Molecular data from living organisms has been used and is being used to inform us on speciation events. How do we get a new species? That's a molecular question. On relationship between living organisms. How are you related to a dog or to a skunk or to an alligator or to a bird? Those things can be teased out a little bit by relationships with DNA and proteins. We can look at the early evolution of venom by looking at the molecular composition of venom in lizards and snakes today. Molecular clocks take some of these changes and trace them back in time, trying to get at the speed at which genetic changes occur in a lineage. And finally, we can look at biogeographic trends and changes in diversity, but we must have the past to do that. That's great for living organisms because there's a lot of them. And molecular information is pretty easy to get. You can harvest corn or you can harvest gophers or other types of animals and just get all the DNA and proteins that you want to study. But for fossils, not so much, and particularly for very old fossils, it's thought that there will be no molecular information. But identification of trends that impact the human species can only be understood through the lens of deep time. If we want to study things like extinction events and recovery from those events, we need the rock record. Global climate change, we can't tell whether it's occurring or not, or whether it's significant or not, or whether it's permanent or not without the rock record. Mechanisms of evolutionary change. How does natural selection work on an organism and how is it retained in a lineage? Acquisition of evolutionary novelties, things like feathers. When did they originate? How did they originate? And what was their molecular makeup before they became true feathers? Those are all questions that we need deep time to understand fully. Factors that influence the retention of new traits in a lineage or an organism and contribute to its success. we need deep time perspectives. Migration patterns, speciation events, all of those things we have to put in the context of deep time. And for that, we need fossils. We need the fossil record. But what is a fossil? Most people think it's one of these things. This is a fragment of fossil bone. Can you tell me that it is bone? Can you tell me which organism it came from? And can you tell me how old it is just by looking at this? That's a really difficult question. But bones and teeth make up the vast majority of what we have in the vertebrate fossil record. And so bones and teeth are okay if that's all you've got, but it does really limit the kind of questions that you can ask. For example, this is a picture of a skeleton of a modern organism. It's complete. There isn't anything missing. If you were to have to reconstruct the animal that goes with this skeleton, what would you make it to be? Would you make it fast, slow, fat? Is it heavy? Is it a plant eater? Is it, what color was it? What appendages did it have? Was it covered in fur or feathers? Those are the kind of questions that are lost without soft tissue. And I bet you anything that if you were told to reconstruct this organism as what it might have looked like in life, you wouldn't come up with this. But that is what it is. It's a camel, a two-humped camel. So as you can see, bones and teeth are enough for a lot of questions. For example, in this critter right here, is it a meat eater or a plant eater? What would your guess be? How about relationships? Is it closely related more to you or to an alligator or a crocodile? Or how about a bird? If you wanted to know how fast this organism could move or what kind of weight it could lift with its forearms, you can probably get at that with just its bones or teeth by just looking at basic physics. But if you want to ask other questions of a fossil like this, you have to have more information. So just imagine if we could get molecular data from fossils, we should be able to use it in the same way that we do for living organisms. And we should be able to ask similar questions of these ancient beings. Why are molecular data important, especially with the study of fossils? Well, basically, all evolutionary change in you, in other organisms that we share the planet with, and in organisms from the past, changes in the genes occur before changes in the proteins, and certainly before changes in their shape, size, and color. The genes change long before the changes are expressed as new traits that we can study in the skeleton of these long dead animals. And although genes dictate proteins, it is the proteins that determine an organism's fitness or survivability. Natural selection does not act directly on the genes, only on the express changes that the genes dictate, but the proteins carry out. So the relationship DNA, which resides in the nucleus of the cells of eukaryotic organisms, has to be transcribed. It doesn't do anything just sitting there in the nucleus, but it's transcribed into RNA and then translated into amino acid chains. These are called peptides. And when these amino acids get modified and get adjusted and get folded into a functioning protein, protein synthesis is complete. In our lab, we use proteins. We focus on them primarily because they're just as informative about organismal relationships as DNA, but they have greater preservation potential than DNA. So most people think that DNA maximum can maybe last 1 million years. Proteins, on the other hand, most people don't argue that they can last for several millions of years. Also, there's a lot less risk of contamination. When you breathe or when you touch anything, you leave behind a trail of DNA, but you don't leave behind a trail of collagen, and you don't leave behind a trail of hemoglobin unless you're bleeding all over something. Also, proteins can be independently validated in many ways not available to DNA studies. And that's really important when you want to detect if contamination has occurred. So for example, if I take a human organism, no matter what I sample, I will get the same DNA sequences. If I sample tissues from their spinal cord or from their eyeballs or from their kidneys, I will get the same DNA blueprint. It doesn't matter. And that makes it pretty hard to tell if that DNA is coming all from one organism or maybe from somebody in the lab or something else. However, with proteins, every tissue is different. The proteome of the lungs, for example, is really, really different than the proteome that you get from extracting kidney tissues. And so if I were to have a dinosaur that had lungs in it, which we don't have yet, and I could get proteins out of those lungs, and then I could get proteins out of their kidneys, these should be different and they should give us different information. And also, that's a great way to tell that, yeah, these probably are parts of the original organism and not contaminants. The problem is things rot, including DNA and proteins. And so it's always been thought, don't bother looking at dinosaurs and other really old creatures because you're not going to find anything. Part of that has to do with how we thought the fossilization process occurred. So what we thought happened was, first and foremost, if you're really lucky, you have a dinosaur that dies in exactly the proper environment. It gets buried rather quickly so that it doesn't get predated upon. And it gets buried, and so it can continue to rot while it's in articulation and nice and neatly laid out. Over time, all of the remaining organics in the skeleton degrade. So the collagen goes away, the bone cells go away, the blood vessels go away, and there's nothing left but holes where these things used to be. And then over time, you have it buried under increasing layers of sediment, and the sediment builds up on top of the dinosaur. Then you have groundwater and snow and everything else moving through, carrying these soluble minerals from the sediment and depositing in those open holes on the bone left by the degradation of this organics. And then finally you have uplift, where people like me can go out into the Badlands and find the fossils exposed. The key thing here is that we can always pretty much tell bone is bone. So this is a modern human femur, your leg bone, and you can see it's got parts. It's got a really, really dense cortex right here. Some of these raised ridges in the cavity that divides it in the middle. And then you've got this trabecular bone, this really spongy bone, sorry. And you can see the same parts here. You can see the dense cortical bone. You can see the lacy trabecular bone on both sides. Bone is bone, or is it? But what if that's not really what happened? What if there's something different going on in actual fossilization? So that's kind of what we were exploring. And to do that, we have to understand what bone is. So bone is a living composite. It consists of a whole bunch of organic stuff. So collagen is the primary protein in bone, but you also have albumin, hemoglobin, and different kinds of sugars. And it makes up what we call the organic matrix of bone. You also have cells, and primarily 3 different kinds in bone. You have osteoblasts, which spit out and secrete the collagen and other components. When it surrounds the osteoblast, that cell changes into an osteocyte. And osteocytes look like this. They're oval shaped, and they have all these extensions coming from their cell membrane that allow them to talk to each other and to get nutrients. And then osteoclasts are an important part of remodeling the bone, chewing it up, and then laying it back down again with osteoblasts to make new bone tissues or to adapt to changes. But these two components together don't make you what you are, and you wouldn't be able to sit upright or take any steps without the mineral that makes your bone hard. And this mineral is pretty conserved. Everybody makes the mineral the same way. It's a calcium phosphate-based mineral. So in modern bones, we remove the mineral so that we can see the organic matrix and cells a lot better, and we can do tests directly on them. But with fossilization, it's thought that these organics degrade away, leaving only kind of a mineral shadow of what the organism was. And if we come along as scientists and dissolve the mineral, we're not going to have anything left, right? And so most people don't dissolve their dinosaurs. They like to save them for museums, but I'm not like that. And we did dissolve our dinosaurs. And we started with a Tyrannosaurus Rex that I had lying around my lab. And I was looking for something in particular, but that's a different story. what I found when we began to dissolve this away, this is a piece of Tyrannosaurus rex bone, and you can see emerging from the surface as the mineral dissolves are these transparent branching structures. And if you look closer, those branching structures are hollow and they've got red stuff inside. So here's a nice branching tubule right there. They're also very flexible. And we studied this a lot and came up with these different kinds of tissues. We have a collagen matrix, which I didn't show here. We have these, what look like blood vessels. They branch, they taper, they're transparent, they're flexible, and they contain little round red structures. And right here, you can see a couple air bubbles, which tells us that they are hollow, but that the vessel wall is continuous and can't leak, which is kind of nice if you want blood vessels. We also recovered cells, and these, as you can see, have the same shape and size as modern osteocytes. They have an oval-shaped body and all these funny extensions coming off of that body. This one is not preserved quite as well, but it's still an elongate, roughly oval shape with little sticky out parts called philopodia, or little feet coming off of the cell membrane. These are classic shapes for bone cells in all vertebrate organisms. And they have to be made of something. If we can pick them up and move them around and collect them, they have to be made of something. They're not parts of our imagination. So when we announced this find, people found it really interesting, but they were also really, really skeptical. This type of preservation was not thought to be possible, and many people didn't believe that that's what we had. So it's true that in science, you can have one example of something and it can be pretty cool, but it isn't really very useful. I mean, after all, one dinosaur with blood vessels doesn't tell you very much. So we went looking for a different dinosaur, and we had a little better idea now of what we were looking for. So this is the side of a cliff. The base of the channel is way down here. You can barely see it. We are working probably 30 meters above the bottom of the channel, and there's probably another 20 meters above us. This is the Judith River Formation in Montana, and there is a dinosaur under here that we're digging out. And when we got it exposed, the bone was incredibly well preserved. So these are the three bones that make up their foot, the metatarsals, and here's a toe bone right here. And then you can see part of the limb going back into the channel that we didn't collect at that time. And we subjected this to the same kind of treatment that we did with our Tyrannosaurus rex. And what we found is when we removed the mineral, we had parallel fibered, white, soft, flexible matrix. That looks just like modern collagen. And we are also able to recover blood vessels and the contents within those vessels, this red gamishy stuff, which is not a scientific word. So that makes two dinosaurs that we got these things from. And so we came up with some hypotheses, since this is science. We hypothesized that these structures are endogenous to the bone produced by the once living dinosaur. If they're endogenous and the dinosaur made them, they should exhibit microstructural features in common with modern blood vessels and cells and matrix. And if they're original, they should contain molecular signals that can be detected by currently available technologies, just like we study modern animals. So in comparing these two dinosaurs, this is a T-Rex. It has this matrix, which if it weren't, 65 million years old, it would be collagen. This is a blood vessel. And inside that vessel, you have these round red structures with the dark center core. In the Brachylophosaur, the second dinosaur that we found, you can see that the vessels are transparent, and instead of having round stuff, they have more kind of a clotted substance inside of them, but it's still red. And when we compare them with modern ostrich and emu, you see the same thing. The ostrich contains round red structures that are blood cells. The emu contains kind of an amorphous reddish stuff and you can also see that these things branch and taper. So the morphology suggests that these things are real or endogenous to the dinosaur. But if I want to study vessels and matrix and these components in modern bone and living bone, I want more information than shapes, because that doesn't really tell me much. And so what we did is we used the specificity and the sensitivity of the vertebrate immune system. So for example, this is an antibody, this black structure right here. It looks roughly like a Y. And on part of the antibody, you've got a structure that is extremely flexible. So much so that if I were to take and inject you with a bacterium I found on Mars, no human's ever been exposed to, I could inject you with that and you would make an antibody to it, to a Martian organism. And when you are invaded by an organism, if this were a cold virus or something else, you can see this is the antigen and it has a whole bunch of different shaped, what we call epitopes on the surface. It is those epitopes that these antibodies bind to, and they bind very specifically. So this antibody with the orange tip will not bind the green epitope. It will only bind another orange one. And so this gives us a way to tell what's there and how it's related to what is present in the cells of another organism. And so what we did was called in situ immunochemistry. So it takes place right on the tissue itself. So we harvested some of that blood vessel tissue or some of that collagen, and we sectioned it very, very thin and put it on a glass slide. And then we exposed that tissue to a primary antibody. This could be collagen antibodies. It could be antibodies against hemoglobin or laminin or some other protein. But we applied these antibodies to it, and then we washed the slide like crazy to remove any antibodies that weren't supposed to be there or hadn't bound. And then when it was all nice and clean and the bound antibodies were still present, we labeled them, we tagged them to enhance what we saw. And then we put a fluorescent tag on that second antibody. The key here is if this little red anti-collagen antibody did not bind, none of this stuff would, and we wouldn't see anything. And this is what we got. This is our T-Rex. Without a primary antibody, you can barely see anything on that tissue. So we use secondaries the same, labels the same, but no collagen antibodies. We didn't see any binding. When we exposed collagen antibodies to the tissue, we see a lot of binding. And then if we take those antibodies to collagen 1 and soak them and incubate them with a whole bunch of collagen, that blocks their binding sites. And so the binding should be reduced, and it is. There's not much binding left here compared to here. And then finally, another control was to test it with an antibody to a protein we would not expect to be present in bone matrix. We used actin in this case, but we've used lots of other ones. So that's the collagen matrix, and that was pretty cool. But what about those blood vessels? What we know in modern animals is that different proteins bind with different avidity and pattern. And so controls that we did were negative. These antibody binding patterns are specific for blood vessels. We used anti-laminin, an antibody against laminin protein that's part of the basement membrane that keeps your blood vessels from leaking out into your tissues. We used elastin antibodies, and elastin gives your vessels stretchability. So every time your heart beats, your blood vessels don't pop. And then we used hemoglobin antibodies because when organisms today die, their red blood cells lyse, and some of that hemoglobin can stick to the vessel wall. As you can see with our dinosaurs, they bound in different patterns with lots of different strength, but they bound the way that they would to modern animals as well. And then we tested the antibodies using, we tested the osteocytes using the same types of antibody antigen interactions. These are osteocytes from Tyrannosaurus rex, and these are Brachylophosaur, that second dinosaur we found. And so when we isolated those, we came up with, if these are cells and not biofilm or other contaminant, we hypothesized that they should be morphologically similar to those osteocytes from modern vertebrates. They should be found in the same environments as those that we see in modern vertebrates. And they should retain some, but maybe not all, molecular features that extant osteocytes also have. So let's look at morphology. This is human bone right here. These are osteocytes still in the bone matrix. They live in these little holes right here. And when you expose them, you can see that they're hooked together. They form kind of a chain or a bucket brigade through these little feet, the filopodia that radiate out. So if I tug on this cell is going to come along with. And that's exactly what we see with our dinosaurs. You can see this chain of cells. They're still hooked together with their little feet. The feet extend out into the collagen matrix. And if I pull this one comes along. So that was good. But that doesn't make them osteocytes by themselves. What we did next was we used scanning electron microscopy, and this is our T-Rex, and this is demineralized bone. So this would be, if it weren't, well, it would be collagen. And you can see these scattered cells on the surface of the collagen and these hairy looking things on the surface. And that looks suspiciously like what we see in modern mouse. This is the collagen matrix. These are the cells, and they're talking to one another by their filopodia that extend out and cover the surface. This right up here is a blood vessel. And if we look a little bit closer at higher magnifications, this is an isolated T-Rex osteocyte, and you can see the filopodia. There is a whole bunch of them, and they're a mess. They make a network of talking to different cells deep in the tissues and more superficial. This one's going down into a minor blood vessel. And again, it's very similar to what we see with a mouse bone cell. So next we go back to our antibodies. And what we show, first of all, is an antibody to actin proteins. Actin makes up the cytoskeleton, the skeleton of each cell. It's a filament protein, and you can see this kind of, the binding pattern is kind of skinny or stringy. When we don't put that actin antibody on and we just do everything else, we don't get any binding. The actin is specific, and it follows this lacy filament pattern that we see in ostrich. The dinosaur, the second dinosaur, Brachylophosaurus, shows the same thing. It is a filamentous protein that fills up one of those cells. There is no binding if we don't use the primary. And it compares really favorably to alligator and how their cells respond to anti-actin. So that's one skeletal protein. We also tried tubulin, and it's a different kind of protein. It's not filamentous, but it does make up part of the cytoskeleton. And this is the T-Rex cell. You can see there's this globular gamish inside the cell, localized to the cell. This is our control, no binding. And this is ostrich, modern ostrich, and antitubulin. The binding patterns and avidity are pretty similar. Brachylophosaurus also binds with antitubulin antibodies and in the same pattern as alligator. So what this is telling me chemically is that these things are very, very similar to osteocytes if they aren't. But the really cool thing was, it turns out that modern birds produce a protein in their mature bone cells, or osteocytes, that nobody else makes. So we used a monoclonal antibody. It only recognizes that one protein. And we exposed Tyrannosaurus rex and Brachylophosaurus to those proteins and saw that they bound like a mature osteocyte from an ostrich. In another test I didn't show you here, it does not bind to alligator, but it's specific to dinosaurs and birds, which we would predict. And it's so specific that this is a chunk of demineralized bone from Brachylophosaur. And when you don't see much here, but when we apply this very specific protein, you can see the cell that is there all along is outlined by that protein. The antibodies do not bind here. They shouldn't. They're cell specific, and that's what we see. So at this point, everybody asked me about DNA, and we did do those tests. This is an antibody raised against DNA backbone. It's not sequence specific. It will bind any DNA that it sees. But the pattern is important. One dot inside the cell of a T-Rex. That means it is not microbial because they have tons and tons of them. Same pattern with Brachylophosaurus, one small dot internal to the cell, just like we see with ostrich. And of course, there's a lot more DNA with the ostrich. A second DNA stain, Propidium iodide, shows the same exact pattern that we see with the antibodies. It is one little dot inside the cell. Same here with both dinosaurs, and a lot more of DNA that reacts to this stain with modern cells. Another DNA test was using DAPI. It's an intercalating stain that gets within the fingers of DNA. Again, it stains internal to the cell in T-Rex and in Brachylophosaurus and in the whole nucleus in the ostrich. Those are all positive for DNA, but I don't work on DNA. So let's get back to proteins, which are fun. So when we tried to sequence the Tyrannosaurus Rex amino acids, we got collagen sequences. We got very specific, undeniably collagen sequences, including one that at the time we did this was unique to T-Rex, definitely collagen. But the machine that we used to get these sequences was pretty low resolution, and it was very unsensitive to compare to what was available later to us. And so we wanted to go back and look at that second dinosaur, Brachylophosaurus. And this is what we saw when we tried to sequence that. This is a much better instrument, much higher resolution, and it yielded all kinds of sequence data. This we know is collagen because of this modification right here. So what we did, we took this sequence that we got and we made a peptide based upon the sequence, and we ran it on the same instrument. This is the synthetic version, and you can see how well the peaks line up. But before you think maybe I contaminated this with my Brachylophosaur or vice versa, the Brachylophosaur has components in it that we don't see in this nice, clean synthetic peptide. So it is not an overprint. As a result, we came up with all of these sequences from Brachylophosaur, and they're all collagen sequences. These are collagen alpha-1 chain and alpha-2 chain. They're different parts of the collagen molecule. And they match sequence identity with different organisms in the databases that we had. When we did the same sequencing with the blood vessels, we were able to recover these vertebrate-specific, eukaryote-specific proteins. So they can't be coming from bacteria. We got actin, which the antibodies also showed us, alpha and beta tubulin, again, that we got with antibodies. We got histone proteins, which rules out biofilm and it rules out any kind of microbe. And then these contractile proteins right here. So we have these by sequence and we have them by antibody work. But the vessel proteins are different than those that we recovered from the matrix. Matrix is mostly collagen. Vessel proteins are not. And that tells us that not only are these vessels likely to be original, like the collagen, but also that they're chemically sequestered. They're not coming in from the outside and settling in these tubes, or we'd find traces of them in the collagen. These data allowed us to build the first molecular tree for dinosaurs. And you can see right here, our molecular data put the dinosaurs most closely related to birds. These are chicken and ostrich. Next, most closely related to living organisms like alligators, but pretty far removed from all of the mammals, which means I didn't put my proteins on there. And that was also extremely exciting for understanding dinosaur molecular makeup. This does not mean that cloning dinosaurs is on the horizon. I think that would be a really bad idea, and we don't do that in our lab. But we're really concerned with what these molecules can tell us, not only about these ancient organisms that we share the planet with, but also maybe a little bit about us. For example, if we look at dinosaurs, they lived in a high CO2 environment. Maybe we can look at their molecular constituents and see how they adapted to changing levels of CO2. That seems to be a problem today. We can look at dinosaurs and say, when did feathers first evolve? How about diseases? When did they first evolve and interact with these organisms? How did dinosaurs adapt to changing environments? We can get a better understanding of evolutionary relationships that are independent from anything that we can get from morphology. We can look again at origin and evolution of diseases. We know that the longer a disease is in a lineage, the less lethal it becomes. And so maybe we can study the progression of diseases in these ancient beings and figure out what that means for us today. We also want to look at the role of microbes in both driving the evolution of these organisms and in preserving them for us to study. And then biomaterials. So we have 80 million-year-old transparent, flexible, and hollow tissues. There's got to be a use in today's world for biomaterials that don't degrade. And these are natural. So I think there's a lot of room for study there as well. But even more so, I think, and more important is once we get past do they persist, once I can quit saying yes, and here's more data, and move to they persist, how can we use them? This is where it becomes important. The potential is limitless. And keep in mind that almost all of the organisms that have ever lived on the earth are now extinct. And that is our eventual goal as well. Extinction. 99.9% of all species that have ever lived are now extinct. So if we want to predict our own future, we have to look to the past. That is the only avenue we have, and the molecular insights into the past are important as well. So that's all I have. And if you have questions, please feel free to email me and I'll do my best to answer them as they come in. Thank you.